All right, welcome to the deep dive, everybody. Today we're diving into something pretty fascinating and something with some potentially huge implications for how we understand our own bodies. We're talking about a brand new AI model called Evo2 and its possible role in the science of anti-aging.
Yeah, anti-aging. That's a big one.
It is. We've been digging through a lot of information, a lot of really exciting stuff detailing how Evo2 works and what it's capable of. And our goal in this deep dive is to, I guess, really grasp how this AI could revolutionize our thinking, revolutionize the way we approach the whole process of getting older.
It really could.
Yeah.
I mean, to really appreciate just how significant Evo2 could be, I think we need to first consider one of the central puzzles in anti-aging research,
Okay.
something called genomic instability.
Genomic instability.
You can imagine your DNA as this complex instruction manual for your body.
Yeah.
Over time, you get little coffee spills on it, pages get dog-eared, the ink starts to fade, and those instructions become harder to read and follow.
Right.
And that eventually leads to all the signs that we recognize as aging. So genomic instability is essentially that gradual accumulation of errors, of typos in our DNA.
So kind of like the wear and tear in the instruction manual over time.
Exactly.
Okay, so we have this problem, genomic instability. We're trying to address this in anti-aging research. How does Evo2 fit into all of this? What are we even talking about here when we say Evo2?
Well, Evo2 is a type of AI. It's called a generative model, and it was developed by the ARC Institute.
ARC Institute, okay.
Now, I think when we hear the words generative AI these days, most of us think about programs that create images like, Midjourney or DALL-E 2.
Right.
Or programs that write text like ChatGPT.
Yeah.
But Evo2, this has a different focus.
Okay.
Instead of predicting the next word in a sentence or the next pixel in an image, it's designed to predict the next building block or the next base pair in a DNA sequence.
So instead of figuring out what word logically follows another word, it's figuring out what genetic letter follows another genetic letter in our code.
Yeah, that's exactly it.
That's a fundamentally different kind of application of AI.
It really is. And the scale on which this thing was trained is absolutely astounding.
Oh, wow.
It was fed 9.3 trillion base pairs of genetic information.
9.3 trillion.
From the simplest bacteria all the way up to complex organisms like us.
Okay, so that is an absolutely massive amount of information to process.
It is, and this massive amount of data allows Evo2 to learn the fundamental rules and patterns that govern how genomes are structured, and it begins to understand, and I'm using that word kind of loosely here, but it begins to understand what makes a functional part of the genome and what constitutes a meaningful change or a mutation.
So having processed all that information, it's developed an almost intuitive understanding of how genomes work, the rules that govern them.
Yeah, that's a good way to think about it. And it's not just a single monolithic entity. There are actually two versions of Evo2.
Yeah.
One with 7 billion parameters and a more powerful one with 40 billion parameters.
40 billion. So what does that mean, parameters?
Well, you can think of parameters like the number of connections and pathways in a human brain. Yeah. So more parameters generally mean that the AI can process more complex information and make more nuanced predictions.
Oh, okay.
And here's another really important point. This whole project is open source.
Wow, okay.
So the data set that it learned from, called Open Genome 2, that's publicly available.
So any researcher could just access this.
Exactly.
That's huge.
It is. And it's built using a framework called Striped Hyena 2, which is also open source. This framework allows Evo2 to look at DNA not just as a linear sequence, but as this complex three-dimensional structure.
Okay.
Understanding how different parts interact across multiple levels. It's kind of like understanding a city requires looking at individual streets, neighborhoods, and the entire infrastructure map.
Yeah, so it's looking at the whole picture, not just isolated bits and pieces.
Exactly.
Okay, so we have this incredibly powerful AI that can essentially speak the language of DNA. Let's bring it back to the big question of anti-aging. How does this tie in? How does Evo2 help us address this issue of genomic instability that we were talking about before?
Right. So we talked about genomic instability, the accumulation of those little typos in our DNA being a primary driver of aging.
Right.
So over time, our cell's DNA just inevitably acquires errors.
Yeah.
While some of these changes are harmless, others can actually give certain cells, or even the little energy-producing mitochondria within cells, a reproductive advantage.
Oh, okay.
They start to replicate more readily, and this kind of selfish growth can disrupt the normal functioning of tissues and organs, eventually leading to the declines that we associate with aging and the onset of age-related diseases.
So it's almost like a microscopic battle for dominance happening within our bodies as we age.
In a sense, yes. And EVO2 has specific abilities that could help us decipher and potentially even influence this process.
Okay.
First, it can pinpoint functional mutation.
Functional mutations.
Not every change in our DNA has a significant effect.
Right.
So EVO2 can help us distinguish between the background noise, the inconsequential changes, and the meaningful signals, the mutations that actually disrupt important biological processes.
So it can tell the difference between the typos that don't really matter and the typos that actually make the instructions really hard to follow.
Exactly.
That seems like a huge step forward because trying to sort through the sheer volume of genetic variations that we accumulate over time must be like trying to find a specific grain of sand on a fast beach.
Yeah, it's a needle in a haystack problem.
Yeah.
But EVO2 seems to be pretty good at finding those needles.
So what else can it do?
Well, second, EVO2 can provide insights into the non-coding regions of our DNA.
Non-coding. So the parts that don't directly code for proteins.
Right. For a long time, these areas were dismissed as junk DNA.
Yeah, I remember hearing that.
But we now know that they play a critical role in regulating how and when our genes are expressed.
Oh.
These non-coding regions act like dimmer switches and volume controls for our genes, and EVO2 has demonstrated its ability to detect functional changes within these regulatory regions, which could be just as significant, if not more so, than mutations in the genes themselves when it comes to the aging process.
So it's not just looking at the genes themselves, it's also understanding the complex regulatory network that controls them.
Precisely. And perhaps most impressively, it has shown a strong capacity to predict the impact of mutations in human genes.
Okay.
In tests involving the BRCA1 gene, which is linked to an increased risk of breast cancer, EVO2 actually outperformed AI models specifically trained on human genetic data.
Wow, that's really remarkable. Outperforming models specifically designed for human genes on a human gene. So let's talk about the future. What does this all mean? What kind of practical applications could we see down the line?
Well, one very promising area is personalized aging diagnostics.
Personalized aging diagnostics.
So imagine a future where instead of relying on these broad indicators of health, we can monitor an individual's genomic drift, the accumulation of mutations over time.
Right.
And EVO2 could be the intelligence behind developing personalized genomic instability scores.
Okay.
Currently, we look at things like blood pressure, cholesterol levels, these very broad indicators of health.
Right.
EVO2 could give us a microscopic, personalized weather report for our cells,
Oh, like that.
predicting storms, disease years in advance.
So instead of a generic approach to trying to age healthily, we could have a highly specific, personalized roadmap based on the changes happening in our own DNA over time.
Exactly.
That's a real aha moment.
And this could lead to highly tailored recommendations for things like supplements, lifestyle adjustments, and even early warnings for specific age-related diseases based on a person's unique genomic trajectory.
So going back to our initial goal, how does this particular capability of EVO2 fundamentally change how we think about tackling genomic instability?
Well, it moves us from reacting to the consequences of genomic instability to proactively understanding and potentially mitigating its effects at a very early stage.
Oh, okay.
And because EVO2 is a generative model, its capabilities extend beyond just analysis. It can actually generate novel DNA sequences.
Wow.
This opens up some really exciting possibilities in the field of synthetic biology for interventions that target aging. So instead of just treating age-related diseases after they manifest, EVO2 could pave the way for preemptive strikes at the very source code of aging.
So like instead of waiting for the instruction manual to get all messed up, we're going in and fixing the typos before they even cause a problem.
That's a great analogy.
Yeah.
We could potentially design new genes or regulatory elements aimed at counteracting the effects of aging at a fundamental molecular level. Think of it as a foundational step towards designing actual youth-preserving interventions.
That sounds almost like something out of science fiction, designing new pieces of DNA to help us stay younger. But with such a powerful tool, are there any safeguards in place? I mean, it makes you wonder, are there any risks involved?
Yeah, and that's a very important consideration. The researchers at the ARC Institute have implemented specific safeguards during EVO2's training.
Okay, good.
It was intentionally not exposed to the complete genomes of infectious disease-causing organisms.
Okay.
And furthermore, testing has confirmed that it is not capable of generating such sequences effectively. This emphasis on responsible development is really crucial.
Yeah, definitely reassuring to hear.
Mhm.
It sounds like the primary focus here is on understanding and combating aging, not on unintended consequences or, potentially dangerous applications. So stepping back for a moment, what's the larger significance of EVO2 in the broader scientific landscape?
I think the emergence of EVO2 really represents a significant move towards computational methods in our understanding of the complexities of aging. It highlights the incredible power of large-scale AI to uncover these intricate patterns within biological data that would be virtually impossible for human researchers to identify on their own. And the fact that it's open source is a real game changer. It encourages collaboration among scientists across the globe, potentially accelerating the pace of discoveries in this vital field.
So to summarize, we're looking at EVO2, this remarkably powerful AI that can not only understand but also generate DNA sequences. It's been trained on an unprecedented amount of data, and its unique abilities to identify crucial mutations and understand the role of non-coding DNA have profound implications for addressing genomic instability, which we've been talking about, this key driver of aging. This could pave the way for personalized diagnostics and even the development of future gene therapies aimed at promoting longevity.
Exactly. EVO 2 really provides us with a new and incredibly powerful lens through which to examine the intricate mechanisms of aging at the genomic level.
Well, this has been a truly fascinating deep dive. You feel like we've only just scratched the surface here, but it really prompts you to consider, think about how the ability to not only analyze, but also potentially design DNA sequences might fundamentally reshape our relationship with the aging process. We could be transitioning from an era of observation to one of active intervention. What kind of ethical or societal considerations might arise as we gain this level of control over our own biology? It's certainly a lot to think about. Thanks for joining me for this deep dive.
Thanks for having me.
It's been a pleasure.
It's been great.